So how do you perform a wound healing assay? So in general, what people do is they first seed a well or a dish with cells, and they will make sure that this well is completely covered with cells. Once the well is completely covered, then you can make a scratch or a wound in this well, for example with a pipette tip. And once this wound is created, you take a picture at the start of the wound, and then also take pictures or one picture at later time points, for example when it's almost closed. And then when you have these pictures, you can measure the width, for example, to determine the decrease in size of the wound. But the width is of course not as accurate as measuring the area. So nowadays most people are using area measurements to determine the decrease in wound size and thus the wound closure. So there are several forms of these collective cell migration assays, and the most well-known forms are the cell removal assays, so where you actually wound the cells. So you're destroying cells to remove them. And some options are, for example, the scratch wounding assay, so that's the most well-known one, but you can also put a stamp on the cells, so you kill your cells by using pressure. You can electroporate your cells using electrodes, or you can even use lasers for laser ablation. So there are a lot of different methods to remove cells. And next to this, there are also assays where you don't remove the cells, but you make sure that they cannot grow in a certain area. So these are called cell exclusion assays. And the most well-known one is the assay where you have a solid barrier. So in this case, you put a barrier over there, you seed the cells, and then you wait until the well plate is completely full with cells, and then you can remove this barrier, creating a gap which can be filled by the cells. And you can do the same thing, for example, with a gel. So there's a gel already in the well plate, you seed the cells, wait until it's completely covered, and then once the cells have completely covered the well, you add a dissolvent that dissolves the gel. You create an empty space and therefore you create an empty space where your cells can migrate into. And the scratch wound assay and the solid barrier assay are the mostly used. So let's focus on them a little bit more. So if we talk about the scratch wound assay, what you do is you remove the cells. And most of the times these are removed manually using a pipette tip or another scratching tool. So this is a very easy tool. You just need the pipette tip, which you already have lying around in your lab anyway, so you don't have to spend any extra cost. However, you can already see in this scratch that whenever you do it manually, a lot of times the scratches are irregular, so it means it's pretty difficult to get reproducible results. So therefore people started to think of ways to automate this process. So there are all kinds of automated devices available which can make very reproducible scratches. However, these devices also cost money, so it is much more expensive compared to this manual scratch method. And some devices that are currently available for creating scratches are the auto scratch, in which this arm moves and goes to each well to create a scratch in the well. Or you have, for example, the wound maker, where you can put a 96 well plate in this device. You can press the lid down and then move the pins, and all 96 pins make the scratches together. So this is a very fast method. So next to this wound making devices, you also have the solid barriers, and you can use a cell culture insert, for example. And what you see is that there's no cells here in the middle, and you see that the cells on the edges nicely aligned with the edges of the culture insert. So there's no wounding of the cells in this case. So this means that you can also get very reproducible images and scratches, but it's also very expensive because you need to purchase these culture inserts, these disposables. And examples of these culture inserts are the culture insert by EBD, for example, where you have two of these tiny wells which you can seed your cells. You also have the ORIS stoppers, so you have a special 96 well plate, and you can put these stoppers in there.